New members. Introduce yourself.
Hi, I'm Ayumi Allred. I'm from Japan and I'm a new member.
Welcome.
Could you say your name again? Could you say your name again, please?
Ayumi Allred.
Ayumi Allred.
Yeah, well, we just joined for our kid who really likes paleontology.
Great.
Yeah, and where's the camera?
Victor, can you turn off the goddess?
Computer is the camera.
Hi, AJ Allred. I'm the dad of this family.
Well, we do have a special guest that we will introduce later, but any other visitors? Well, we are glad to have everyone here today. I would like to introduce our very special guest speaker, Dr. Patricia Gensel from UNC is going to talk to us today about fossil plants. And I met her first myself in 1998, which is kind of way back, and did a seminar at NCAT with her, and then a couple of times ran into her in the field. So I'm really excited to get to hear her speak today, since it has been a while. And so I want to say welcome, Pat, and we're looking forward to your talk.
Well, thank you. So yes, I've been around for a while. So how many of you have gone on a field trip with me? A few of you. Some of this may not be news to you. I wasn't sure of things. So I actually want to start with some questions. So first, I'm very pleased to join you. I'm just sorry, I'm really kind of chicken still about COVID, so it's remote, but let's see what we can do. So my first question I have for you all is, what comes to your mind when you think or hear the word fossil? And anyone can answer. You can unmute and answer, those who are virtual or those in Zoom. What comes, just think what came to your mind when I said the word fossil?
Ferns.
Yay. Anyone else? Ancient.
Dinosaurs.
Dinosaurs.
Oh no.
Uh-oh. I see a smile and wood. That's cool. Anybody say, okay, where, when, and what?
True. Yes.
Okay, so I have a second one. What's your favorite fossil or favorite kind of fossil?
Invertebrates.
Particular kinds of invertebrates?
Marine invertebrates, because I grew up near them.
I second that. But fossil wood is fossil wood is a close second.
Have you found much fossil wood?
Yes.
I need to monitor this chat. I think it's looking very good. So we got prehistoric here. What are slime queens? You're going to have to inform me. Oh, Cretaceous vertebrates we've got. Anybody for shark's teeth or something simple like that? Or is everybody doing other things? Do many of you have a collection of fossils?
Yeah. Oh, yes.
Any fossil I find, I'm with you. Yes, find collections. And so in a sense, I wanted to get a feel for what people's interests are. I work with fossil plants, and I work with really ancient ones, and so they don't look a whole lot like anything that is living today. They're mostly extinct now. Some of the major lineages they belong to are still with us. But if you were to be able to time travel back to 400 million years ago, which is the age of the fossils that I work on, the world would look very, very different than it does now. So that just for an introduction. Now I'm going to share my screen and start what I have. And so I have a couple of goals here. See if this does it. Can you see my slides now?
Yes.
Yes.
Yeah, can you put them in presentation mode?
Oh, I plan to as soon as I do
Okay,
that.
thank you.
There you go. How's that?
Perfect, thank you, looks good.
Okay, so I changed the title a little bit from what you may have had, but I'm not sure what the right title for this is. But in a way, what I wanted to do was to first express appreciation to all of you who love
Saw
fossils.
this on Facebook.
Pardon?
Kristen from Kristen. Noah's 5th birthday. All righty, we are less than a week away. So if you can let me know if you haven't RSVP'd yet,
Excuse me.
that would be great. Thanks, guys.
Here, can you tell people
What?
tell them to mute?
Oh. It would be helpful if you would mute if you're not talking so we don't get feedback from your computers if you're on the Zoom.
I'm gonna mute people.
But if you have a question, one thing I'd like to say is if you have questions as we go through this, please unmute and ask, or if you're in the audience, shout it out. So we're going to look at, I think, a couple of things. How I, as a paleontologist, go about studying fossils and how that might compare to what you guys do as people who love fossils and collect them too. And you know, some of the kinds of techniques that we can bring to bear to understand things. And also that there's some really big questions, which I'm sure you've thought about too. And so we'll try to touch on some of these things as we go through. So first of all, when you think about fossil plants, those of you who love dinosaurs or other kinds of vertebrates could imagine that plants are actually the background and the surroundings and also the habitat that's been provided in which those animals live. So plants are really central. They kind of define ecosystems or vegetation types or even biomes. They reflect things like soil, moisture, climate. They're also a primary producer, which means they're a source of food for most heterotrophic organisms, those that have to take in carbon to build energy. They provide oxygen to the atmosphere. They do all kinds of important things. And if we start from when plants are first known to occur in the fossil record, we also can see that they changed essentially the surface of the earth. They helped to form soil along with other kinds of organisms and also altered the general environment at that time. So as a result of over 100 years of work in different time periods, we can actually show and feel moderately good about some of these environmental reconstructions that are focused mainly on plants. It'd be fun to put, there's a couple of animals in this one, but I don't think I got some in some of the others. But so I put up here what the vegetation might have looked like during the Devonian period of time, which is a time I spend a lot of time investigating. And it was probably more like a marshland, very small plants, or a late Carboniferous swamp forest. This may be something you're very familiar with the giant lycopod trees. Here's the cast of a base of one of those. These things got really, really tall. This one, I think, is about 5 feet across, at least in diameter, right about this level at the base of its trunk with these big spreading roots. And they're the yellow ones and the blue ones that are scattered around in this reconstruction of a swamp forest. Then some other forests we have, these are some of the earliest rainforests that people have talked about, would be the Paleocene or earliest tertiary rainforest from the Denver Basin, and then a late Cretaceous one, what immediately preceded it. So this brackets the major extinction event at the KPG boundary and shows plants before and plants after. And plants did recover within a reasonable amount of time and didn't take quite as bad a hit, perhaps, as some others. So when we're studying fossil plants, we have all kinds of questions that range from what is it to what can I learn from it, and who else is there with it, and what kind of, ultimately, if you can work on enough plants from a site and you can understand the geology of it and know whether they've been deposited in place or transported in, you can actually build some idea of what that paleo environment was like, what the plants were like, and even what animals might have existed with them. So I've focused a lot of my time on early Devonian plants from several localities in northern Maine, up in northern New Brunswick, and also on the Gaspé Peninsula. And in all the time I've collected plants, we found a few arthropod remains, little things that probably are similar to the roly-polies that you find outside today, and one fish. So I haven't, but there are other fishes in various places as well. So we do encounter some other organisms, but not always interspersed with the plants. The New Brunswick deposit also yields one of the earliest known sharks and a variety of fish and some invertebrate types such as ostracods and a few little early, probably not yet clams, but molluscan type things. Okay, so when we look at plants and we want to understand them and perhaps extract information from them, what we can learn depends on how they're preserved. So a lot of them are impression fossils or imprints. This may be review for you. You got to tell me if it is. Does everybody know all about this stuff?
No.
No? Okay.
Not everybody.
All right. So, okay, just enjoy. So this one is mostly an imprint. It has iron oxide stain that gives it some contrast to the rock. This one, in contrast, has some of the actual plant preserved in the rock. It's been compressed, it's been subjected to pressure and cooked. And so what we get is a carbon film over resistant parts. And the resistant parts on plants, one is the outer covering of a plant, the waxy outer covering that we call a cuticle, which can yield a great deal of information. Another would be any resistant thick-walled cell that contains material like lignin, or in the case of its reproductive propagules or spores, sporopollenin. These are very, very tough, high molecular weight polymers that can withstand a great deal of heat and pressure. So with this compressed material, I can actually either pick the plant off the rock, or sometimes what I'll do is digest it in acid. So we use a mixed first hydrochloric and then hydrofluoric acid. So it's not, they're not nice acids, but we can do that, break down the rock and remove the plant pieces. So what you see in these three images right here, this would be a uncleared piece of stem plus a sporangium. This is the structure in which meiosis takes place and yields spores that start sexual reproduction. I can get spores out of those sporangia. Here's a stem again, and you notice there's spines. And in this one, if you look at it, you can tell that the spines are occurring all around the stem. So there's a great deal of value in being able to extract things from the rock and look at them, be able to turn them over and manipulate them in a dish. Now, the other major form of preservation we have would be permineralization. And in this case, you might know it as petrifaction, but we tend to make the distinction that when something is petrified, all of the organic material has been replaced by mineral. In this case, we still have organic material present. So if you look at this one, everything that's sort of black, the cells around the periphery of the stem, called the outer cortex, and the cells in the middle, these are all lignified cells and very resistant. This is the water conducting cells, or the xylem. And while this has all been impregnated, in this case with limonite, which is a breakdown product of pyrite, there are plant cell walls embedded in there, and that's what's remained on this thin section, which was prepared by grinding and etching with acids, or polishing in other cases. Other things can be preserved in silica or calcium carbonate, and usually we can actually find that there's actual plant cell wall material that is just been infilled and also surrounded by these different minerals. So with that, we can make sections either transverse to the long axis of the stem like this, or along the long axis and build a picture of what that plant is organized like. Okay, so actually this is kind of what we would start out with. So if you look at this, this is from the Devonian. What do you see? I'd like to have a couple people's impressions here. What do you think you're seeing?
Carbonized plant stems.
Carbonized plant stems. You've been doing this a while. That's right. Right here and here. I heard somebody else say something.
Yeah, plant stems.
Okay. And what are these stems doing? Anything? Do we just have like a plain old stem that goes up and down, or is there something?
Well, they're all aligned in the same direction.
They're all aligned in the same direction. So that
would say water flow to me.
Exactly. Most likely water flow.
And they're branching.
Yeah.
But the alternative to water flow was that they all were growing in the same place and got buried right near where they grew and just got knocked over.
Yep, by wind or water or anything. Yep.
Right. So there's a couple of things you could do with that. But you mentioned branching. So yes, you can see that there are places where these stems branch. Now on this one over here, I'm going to draw your attention to these little structures right there, because those are kind of important in helping us identify what group this particular stem belongs to, because what those are some of those sporangia, those reproductive structures, and they're elongate. I'll show you better ones in a few moments, but so there's some surface information that you can get from here. Now this one's been possibly prepared a little bit. Can you see the white lines along the side there? So I think what we've done is to take a fine needle under a microscope and chip away some of the rock to expose portions of the stem or the sporangia in parts of this specimen. This one I don't think we've done much with yet, but we could do the same thing there and expose more of each of these stems. So another question you might ask about this fossil is, okay, you got all these stems all lined up in a row. Is that one plant or is it several?
Could be either.
You're right. So it could be either. We can't tell. And that's something that's a little frustrating, but we have to deal with what we get. And I think the challenge for all of us is to learn as much as we can from the fossils that we can find using whatever techniques come to hand. Now here's a close-ups of that plant. It is a Xylophyton. This is one of the iconic genera of early Devonian plants. I've described a few, I have more to go. This one shows some of these stems that branch, and here you can see lots of these sporangia. In fact, here's a cluster of them, and obviously the branch is not exposed, and it's either on the counterpart of this rock or going down in the matrix. Here's what it would look like when it comes out, and those branches bifurcate a few times, and then you bear the sporangia. Here's a piece of the Xylophyton that was removed by digesting an acid. This was buried in a volcanic ash up in New Brunswick, so it was extremely well preserved. That's probably maybe about 3 to 4 inches long, and that's pretty good sized piece to get out from acid maceration of that type of thing. And there's a lot of branching going on. And this particular Xylophyton has spines and a few sporangia up at the top. This is an image of a sporangium after it's been cleared. The carbon has been cleared away. You can see it splits open along one side like this. These are all features we use in these very simple plants to help recognize and distinguish one from the other. And it splits open to shed spores. So all these little dots you see, the magnification isn't big enough to tell you, but those are all spores, the little reproductive propagules that would be shed from the sporangium and grow into a haploid plant that gives rise to the next generation. And then some of these Xylophytons are permineralized, not this one or that one, unfortunately, but others. So this shows, again, a Xylophyton with its vascular bundle and a cortex, and then this one, the base of the spine. So this one was preserved in calcium carbonate, where we could actually make a special kind of thin section called a peel. I don't know if any of you have done those, but they're fun, where you just etch the surface and then you put down acetate paper, let it harden, and the cell walls standing in relief get embedded in the acetone. You can peel it off and have an ultra thin section. Okay, so when we get done looking at any one of those kinds of silophytons and studying all the specimens we have, we can make a reconstruction. So here's 3 different species of silophyton. And you know, one thing that you notice, it's sort of truncated down here and down here. That is, how much of the plant have we got? So far what we've suggested is that silophytons are plants that perhaps are somewhere between 1 and 2 feet in height. Each of these stems is about 5 millimeters to 8 millimeters across, depending on which species you look at. And then as you get to the more distal branches, they become narrower. But do we have the whole plant, or are we missing the base? We're definitely missing the base, but how much of the base are we missing? And so that's something that I have a project kind of waiting to be looked at, where I think I might have some more basal regions, and that these might have been just the distal portions of much bigger plants. So these are some of the things we know, and there's much yet to be learned. So maybe I should have started with this, but you now know something about silophyton, and it's very typical for early Devonian plants in many respects. And so what we can say, if we thought about all of the early Devonian plants we know about, is that they're small, comparatively small. They lack any well-defined roots. We've never found any. Sometimes it's because the bases are missing. Other times because there's apparent, you know, we just don't see anything that could be rooting structures. They apparently lack leaves, and so they're quite different from some of the plants a little bit later in time. I wanted to circle, I had a circle here, come on, do it. There we go. So what we're really looking at is this period in geologic time where life began here. Now, if we were to go back as far as this, the plants I've shown you so far are about 400 million years old, but we do have evidence that plants existed as long ago as 450 million years in the Ordovician. And it may even be earlier based on some evidence that is beginning to come out. And most of this is just their spores. So we don't know anything about those pre-Silurian plants. For example, it's really from the late Silurian into the Devonian. So a Silurian plant is shown here, Cooksonia. Notice the scale is very different. So this is much, much smaller than Silophyton. But you know, there are bigger ones in this late Silurian as well, which would suggest, you know, we've got plants very much earlier than the megafossil record, meaning whole plant or parts of plants like this have been discovered. Okay, I'm going to continue on the Silophyton theme for a while, because like I said, there's more to be learned. We have anatomy, but what we've had available for anatomy is oftentimes just very short lengths of a stem. And I didn't mean to jump ahead, but when I cracked open this rock, it was pretty exciting because we've got somewhere in the range of about 8 to 10 centimeters worth of permineralized stem here. It's all preserved in pyrite, and we've got part and counterpart, so we can put all those things together. And I set this as an undergrad research project for a student called Nick Chamberlain, and he made beautiful thin sections, polished thin sections of the pyrite. So you can see some of the processes of the stems undergoing division shown in this sequence of slides right here. So it begins here, it's pinching off there, separating more there, pretty separate here, and then they become totally separate. And here in this one, I've jumped, they're not all exactly one after the other, but you can see that it divided a couple of times. So this is a very laborious process. You have to embed the specimens, you have to make the sections and keep track of them, then you have to etch them or polish them, etch them, and then mount them on slides, and then you can take the pictures. So Nick lined up a series. So here we go, front and back of polished sections through a sequence that shows similar kinds of division. And this is a good way to show something about what happens over the length of a stem and how the vascular tissue might change when division is taking place. But there's another way, and this happened after Nick did all that hard work. So let me see if, oh, it's not going to work for me here. Come on. There should be one right here. There we go. So watch this. So this is called micro CT scanning, or X-ray tomography type scanning on a micro level scale. So what we did here was to take some specimens, put them into this micro CT scanner, scan them, which took somewhere in the range of about 8 hours. And it created extremely thin sections, all digital. It's like getting a CAT scan of a 5 millimeter, 1 inch long piece of fossil plant stem. This approach has been used on fossil fruits, on dinosaurs. I did this in collaboration with Selena Smith at the University of Michigan. And then you can actually either look at just the thousands of thin sections that you have, or use a very expensive piece of software and actually be able to visualize it. So this is visualizing it in one axis. And this is a movie just showing what happens as you progress vertically through that series of sections. Now here, we're going to look at it going from a different angle. And again, you start seeing some things. Now before, you might have felt, okay, you had a central axis and then you saw a couple things and you assumed that maybe they'd been produced by that central axis at about that time. But when we looked at it at this angle, you notice this right here. Here's these laterals. If I move it along a little bit further, like so, you can see them coming off. So that's actually a dichotomy that took place way down here, not up here. And so another thing you can do with these digital sections is you can generate a 3-dimensional reconstruction and then slice it any way you want and look at it in any way you want. So I think this is the wave of the future. I did this just before COVID, so I haven't gotten back to Michigan to do more. And I don't have my own micro CT scanner, although I would love it, but you know, that takes quite a bit of money. So I'll wait until I can get back to Michigan to do a bit more. Okay, so that's a cool technique that I think there's more room for work on. All right, so there's other plants. Is there a question? Are we good? Okay, so following up on other plants that look like Xylophyton or even are associated with Xylophyton, one of the most exciting finds that we had occurred over a decade ago, but it's still kind of exciting. And that is that we found that some of these early plants, instead of having a tiny little vascular bundle in the middle, had a much bigger one, like you see here, with 2 zones in it. There's a central zone and then an outer zone of very aligned cells. So somebody mentioned that they really like fossil wood. And so this is wood. And to identify something as wood, you need to meet certain criteria. So we looked for this. So this is a plant from the earliest Devonian of France, beautifully preserved in pyrite, where we could actually make peels of the pyrite. And so one thing you look for is this alignment, and then where you see one row splitting into 2, where what we call doubling, and where the yellow arrows here show, here's the 1 row and it becomes 2 rows. The other thing you look for is the presence of what are called rays. So most of the xylem is going up and down, but ray cells are going across. And we do think we found them in both of these, for instance, in this zone right here, and in this blank space right there, because rays don't have resistant walls and they wouldn't likely get preserved. But whatever, we think that even a 5 or 8 millimeter stem had the capability in some plants to produce a certain amount of what we call secondary xylem or wood. And that this was a great advantage to plants, because when we look at wood in living plants, it provides both 2 things. One is more cells to conduct water from the base of the plant to the top so that they can get larger in size and taller and also live longer. But second, it provides support. So it's kind of exciting. Before this, the earliest evidence of any wood was about 10 million years or 15 million years later in time. So this pushed it back in time a great deal. So it's sometimes I've talked about Devonian plants and finding things happening earlier and earlier, and this is a good case for that. Now this one's interesting because these are axes affiliated that look like they're silophyton. I'm still trying to put all the pieces together, but that too has what appears to be wood. And if so, that could be what the more basal region of silophyton contains. So it might have wood in the more basal regions and less or no wood at the top in the younger parts of the plant. Okay, so I want to show you a few other Devonian plants where we have learned some things, but have more. I'm still getting a little background. Is that just in the room? Okay, so in this case, these are the same age as that silophyton, but they're much, much larger. So this one would be, in my estimation, somewhere in the range of 4 to 5 feet tall in life. And we only have a part of it, and it's incomplete at both ends, and it's reconstructed here. Here's another one about the same size. So these axes are about 1 centimeter across, have these side branches. In this one, they're very short and equally dividing multiple times and produce tight bunches of these same kind of sporangia. In this one, it produces smaller, looser bunches on dichotomizing axes, but then the side branches get a bit longer. The trouble is we don't know anything about the anatomy of these plants, and it would be interesting to know that. Now, I have a feeling that it's some kind of a very deeply lobed vascular bundle based on a plant similar to this one that I'm studying from New Brunswick, but bigger guys than the others. So we might have had 2 tiers of vegetation in terms of height in the early Devonian in certain areas. We also have a group of plants in the early Devonian called the Zostrophils. Several are shown here. You already kind of have seen these. This is Saudonia. This one is from a plant called Crenaticollis. All of them have these laterally borne sporangia. Some have smooth stems, some have spiny stems. One question could be why the spines? And there's several ideas that somebody could put forward. Why are plants spiny today?
Protective mechanism, so they won't get eaten?
Yeah, so protection against herbivory is one. Our problem is we don't know who the herbivores were. And we don't have any. We have these arthropods, but they're lacking their mouth parts. And from the coprolites we see, there were some things that feces can actually contain things like spores or remnants of beat up cuticle. So there's indirect evidence, but not direct evidence. So that's one distinct possibility for this. Another one might be that it changes the boundary layer of air. So if you think about air moving across, and then these plants might be living where, they don't want to dry out too much. So you'll find that plants that are living in very windy, exposed areas often have either hairs or spines all over them. And it moves the boundary area out to the outer tips of the appendages instead of right along the surface where all of the stomata are, the little pores where gas exchange takes place, so that it would cut down on water loss. So that's another possibility. And we simply don't know the answer, but my guess would be maybe both. These are an interesting group of plants. There's probably somewhere in the range of about 20 or more genera of the Zostrophils now known from the early Devonian. And a few of them hang on until the end of the Devonian period of time. But this is what we think their growth habit was like, that they had creeping stems and some aerial stems on which they bore their sporangia. I'll come back to them in a moment, though, because we might know something more about it than that. So another group that we have are the lycopods. So how many of you are familiar with things like running pine, ground pine, princess pine today? Anybody know? They're little creeping plants that grow in deciduous land. Okay, so this is an ancient group. Those are lycopods too, not pines. Somebody thought they looked kind of like pines. So here's some Devonian ancestors for some of those guys. And this one called Drepanophycus has leaves and then sporangia that are born on directly on the stem in between its leaves. And this one's interesting because this part is spiny, that part's spiny. These things are not. And so we suggested these might be going down into the soil and functioning as roots, whereas this was the creeping stem on the surface. This comes, by the way, from Bathurst Island, which is located about 65 degrees north up in the Canadian Arctic archipelago. And in the Devonian, it was probably sitting at what was then the paleo equator. So we can find plants even up in the high Arctic, and you'll see that again later too. Here's another lycopod, Leclercia, and we don't know what its growth habit is because this might be the most that we get for the plants, but it had forked leaves. They have 5 prongs to them when you can put them together. But in this case, the sporangia are born on the leaves, which is a typical feature of all lycopods from that point on until the present, even today. Sporangia are either born on the leaves or in the axils of the leaves. And they also have, I think there's several things here that talk about some of the characters that help recognize them. But here's some early members of that group. And some of these actually go back into the late Silurian. So the lycopods have been a separate lineage of plants for a very, very long period of time and have produced, especially in the early times, an herbaceous component to the vegetation, creeping plants that didn't get too tall. But we're going to see they became tree-like, and then they became herbaceous again. So it's kind of an interesting story when you look at it across geologic time. All right, so we also think we see potential rooting structures in some of the zostrophils. This is Bathurstia from Bathurst Island in the Canadian Arctic, and another zostrophyllum from there. And these structures pointing downwards, and this is upwards with sporangia up in this area, can only be roots. Now, the interesting thing about these plants is that they represent plants that essentially got ripped up from where they were grown, probably as a clump, and rafted out to sea, because the geology tells us these things are 200 miles out to sea. They're intermixed with graptolites and other kinds of marine invertebrates. And actually, it makes it interesting when we were looking for them because we think, oh, there's a plant. No, it wasn't. It was a graptolite. If you know what graptolites look like, they look like little bitty skinny saw blades. And so we got faked out half the time when we were trying to find plants. Oh, there's a bloody graptolite again, but people worked on those too. Okay, so those are the main groups. There's many others, and I could spend some time on that, but I wanted to kind of do something else. So I'm going to kind of hit a few high points that we actually have things with leaves as early as the late Silurian. Here's our plants with rooting structures. There's also some maybe back in the Silurian, but especially in this period of the Devonian. And we have woody plants at that time. Now, we couldn't put all the events on one of these timelines. So I'm showing you a second one that shows earliest plants in the Ordovician. I want to point out some trees in the Middle Devonian because I'm going to talk about those next, and seed plants in the end of the Devonian. So we can say that most major events in the evolution of plants and plant structures took place from roughly, especially the late Silurian through the end of the Devonian. And the only thing that's missing is a flower. So we got everything else, but we still have this very, very different kind of vegetation, but there's tons of innovations that have gone on during this time. So is everybody with me or have a question at this point? Because I'm going to tell a different story now. We're good? I hear nothing.
We're good.
We're good.
Okay. All right. So what I thought might be fun to do is talk about forests. And in a sense, I'm an onlooker on this. It's my colleagues who've done this work. I have reviewed their papers. I've encouraged them. I've seen their sites and things like that. So this is another thing that, say 20 years ago, we didn't even know existed, where was forests in the Middle Devonian period of time. So this is only a few million years after things like Xylophyton and Pertica. Now, one frustrating thing when you're trying to track the history of these early plants from the early to the Middle Devonian is that there's the earliest Middle Devonian, the Eifelian, there are very few fossils known. And from the end of the early Devonian until the Jivetian, the next stage in the Middle Devonian, a lot of change must have taken place because the Middle Devonian plants we find are really very, very different. Now there's little ones still, like in the size range of Psilophyton, of different affinities. The lycopods are still around, but some of them, while they look like early Devonian forms, also look very, very different. So what we do find, and what has been exciting, are some of the earliest known forests. So actually, a good question is, how do you define a forest? What makes a forest?
Trees.
Well, trees, that's right.
Grouping of different kinds of trees that help each other.
That's a possibility. I'm not sure I know a good definition of a forest, but first of all, a group of 6 trees doesn't make a forest, for example. You need something that's covering a decent sized area. And certainly one thing we see within forests, and what is it called? The Mother Tree is the recent book about some of the forests in the Pacific Northwest. We know that trees interact with other trees when they're growing together in a forest by means of oftentimes fungal symbionts in their rooting systems or sending chemical signals through their roots from one to the other. There's a good chance this might have even happened as far away long ago as this. So the Middle Devonian, about 375 million years, we actually find trees, and they range in size from something that might be about 6 feet tall to things that are probably 30 feet in height. So some of these have been known from parts for a while. So for example, these stump casts have been known since the 1920s. And some others of them have all been discovered within the last 2 decades in order to put this together and be able to talk about forests. So there's big words here that you don't need to worry about, but this one is the thing called Eospermatopterus, because that's what this stump was called. And others we'll look at later as Protolepidodendropsis. And then these are actually over here reconstructed, they're lycopod, but these formed forests. And so the big question is, how can we say that? And so I want to tell some stories here. So some of the forests occur in New York State, some of them occur in Germany, and some of them occur in north of Norway in Svalbard. And there's also now one in China, and I suspect we'll find more because now we know what to look for. And it involves a lot of excavation. I mean, if you go and approach an outcrop from the side, it might take you a while to recognize something as a forest. And I'll come back to that one too. So actually, paleontologists from the New York State Museum in Albany were kicking around in the Catskills and looking at quarries, and they came across a quarry where they found evidence of this cast of a tree that was lying right across the floor of that quarry. And so they marshaled their forces, they put everything in, they took it apart because they couldn't take it home all in one piece because it's 30 feet tall. And they encased the casts in plaster Paris, put them on a truck, took them back to the museum, put them together, and they photographed things in pieces, stitched it together so you could have a view of this tree now that it's removed from its quarry where it was lying. And here's a reconstruction of the parts that were found, including the aerial parts. Now, one critical thing about this tree, you can't see it in detail here, I'll show this in a moment, is that it doesn't have leaves. It branches until it has very fine little divided branching like things at the tip. So probably all of the branches photosynthesized maybe the younger parts of the stem. We also know because there's scars that the branches dehisced. So I'm going to jump ahead for a moment, a couple. Here's some more pictures of this plant. So here's a portion of its stem. This is the upper part where the branches are going off at the top, and you can see these things where branches have abscised or departed from the stem and fallen off. And here's one where, it shows it in the outcrop where they got the branching portion of it. There, one time years ago, I actually saw a tree in the Upper Cretaceous in the Neuse River cutoff, lignitized fossil tree that extended all the way across that river. And so I think this would have been a comparable kind of experience, except they were able to do more with it. Okay, so you have also in Germany a number of complete trees, not like this, but more like that, but smaller. And then in Svalbard, this. So we have forests, and I'm going to kind of show you a little bit more about doing paleobotany on a macro scale. I admire this. I think this is pretty cool. And some of it came about by chance and luck, but some of it came about by just going out like all of you do on collecting fossils and finding things, and then persevering in getting more information about it. So this is the quarry at Gilboa, New York, which is in the Catskills just north of New York City. It's a quarry where they mined rock to build a dam to make a reservoir to provide water for the city of New York. This was done in the 1920s, and that's when they found those stump casts. However, maybe about 15 years ago or so, they started opening up that quarry again and to get rock out to repair the dam. And when they did, some of the museum people plus Bill Stein from Binghamton University in New York went and they found the remnants of a forest floor. So this thing you see right here is actually a soil horizon that represents the ground level of a forest. And it's got water in it right now because they stripped away all the rocks when they discovered it. They had help from the DOT in New York State to do that. And then they also had to drain out all the water. But what you could find is like, this is one of those round circles that you can see, for instance, here, or there, which this is where those stump casts were removed or existed. So this is the base of one of those trees with the rooting systems extending out like guy wires from all around the side. So that's a root mound. So one thing that they did was to set up a grid, and I'm going to jump ahead again a little bit, gives it away, and they gridded it off. You can see the lines here, and they set up cameras, and at night they took polarized pictures of each of the grids, and then they stitched them all together. And so the first one they did is this forest here, so they could talk about the size and spacing of the trees that made up that forest, and also the kind. So this is the Eospermatopterus tree with its radiating root system. You notice there's big ones, and there's also little ones and middle-sized ones. The light gray are trees of a different kind that they haven't identified. Then these black lines represent a vine-like plant that is a member of a group called the Progymnosperms that's creeping along the forest floor, comes right up to the trees. So they think it grew up the trees like grapevines do today. And so, they were able to work out, here's a forest without a doubt, and something about the density of the trees in that forest. The only thing that's missing are the trees themselves outside of their bases and the casts that have been removed with earlier quarrying operations. But that helped them put together this Eospermatopterus type of tree. So this is one of the forests. Now, a second one is at a town not too far from Gilboa called Cairo, New York, and there's another quarry. And what they found here were rooting systems that radiate out. So all these white lines represent a rooting system of a big tree with probably this bucket representing where the trunk of the tree was. And this is more similar to the kind of rooting systems you see in oak trees or sweet gums or various pines today, right? It's kind of radiating out like a lot of Gymnospermus and Angiospermus trees do. And there were several of these. So they employed the same techniques initially. And then along came drones. And so after having done all this laborious photography, they actually bought a drone and were able to kind of refine their work, again, gridding out everything. And to look at the composition of this particular forest floor. So here are these trees, they belong to another progymnosperm called Archaeopteris, which is a large tree with these radiating roots, but it also had some Eospermatopterus in it. So this is the second fossil forest. Now, one thing they're doing now, because one thing we're interested in is how have plants affected, say, Earth and Earth system processes and soils, is that they have taken and drilled cores, both at Gilboa and Cairo, and determined the thickness of the soil and are doing geochemical analyses to understand what type of soil it is and what types of climatic conditions it tells us existed at that time. Some of that data are still forthcoming, but I think this is pretty fascinating for not too long after the plants I work with, like Xylophyton and Pertica. Now, the last one, if you go way north of Norway, practically, it says 70 degrees here, 79, I'm sorry, 79 degrees latitude right here. You come to Svalbard, which is sort of a broken up set of islands. And there's a lot of fossils on Svalbard, beautifully exposed and not covered by vegetation, obviously. And when they were, a couple of my colleagues were working there, they found, again, casts of stumps, in this case of a lycopod. So these things that look to you like drill marks are actually stump cast cavities, you know, the molds where the casts came out. And they were actually able, once again, to map these a little bit. So here, looking down on them, there's these stumps all over the place. Here you can see what they mapped out. And if I do this right, what they were able to do is to tie it to an already described plant called Protolepidodendropsis, which is a tree-sized lycopod about, well, what does that say? This is, it's probably about a meter and a half. So that's about 8 feet tall, something in that range, and it branched at the top. We know of other tree type lycopods in the Middle Devonian, as well as in younger sediments as well. So we have now several early forests that predate what, say, in 2020 would have been considered the oldest forest made of Archaeopteris. So they go all the way back down into the Middle Devonian. So there had to have been a big change in vegetation in that time. Okay, so I kind of wonder now, I've worked on this outcrop for years. Some of you have been there with me, and including a member of your audience who waved at me earlier. And I think we've been working on a forest and didn't know it. So this comes from the Lower Carboniferous in southwestern Virginia. And fossils accumulated from there. Some of them are kind of casts, flattened casts, as you see here. So this is about a 10-foot long piece of stem that was put back together after we pulled it out of the outcrop. A student did this actually, with some details of what that looks like. And then this probably would have attached to the top of that and it forks. And then this attaches to one of these and it forks. And that attaches to there, and this attaches to this. So it had a crown that branched multiple times. If you look at the surface, it has scars where leaves were born. We have the attached leaves. And it originally was called Lepidodendropsis. But one thing that my colleague Kathleen Pig and I found when we were describing this is that it has features inconsistent with calling it that. And we didn't give it a new name, but we still need to decide. So actually it's going to become Rickyphyton, but it's another tree like Hopsid. And if we look at this next series of images, this I've seen several times, but I think we really hit some good stuff when I joined Jim Mickle in one of his paleobotany classes on a field trip to Virginia several years ago. And this still needs to be, worked up. We've known about the bases of that tree, what I'm going to call Rickyphyton. That's not been published yet. So these are, these casts occur all along, like in here and here and here, all along here you could find them. But the trouble is we don't have even sediments with a forest floor. What we have is a forest that's been jostled about by mountain building so that, we couldn't do the same kind of thing that has been done on those earlier forests. But this is certainly an aggregation of tree lycopods of a certain type. Now, these differ from the upper Carboniferous ones because they probably had lobes at their base, and you can see these lines coming out all along here and here, lines coming down from each of these. So these had these guy wire types of roots coming out from their base. And that we found in great detail with Jim and his class on that field trip. So that still needs to be worked on. But this could be a forest as well. What do you think, Jim?
Sure. Yeah, let's see if we can find that. Yeah.
So
that's exactly what we need to find.
Yeah, I think it's pretty much, I mean, it's a more limited view of a forest, but I think it's a forest. So now I think what's interesting in the Virginia stuff is that we have just adjacent to where all of these stump casts are, the lithology of the sediment changes, which says the depositional environment was different, and we get out foliage. We go up the road and around the corner, and we found this foliage type, which I'm currently trying to write up with an undergrad who did work on it. And this one looks very much like something that's been known in Europe, but it's a little bit different, just like a lot of our Virginia fossils are different from those known in the lower Carboniferous of Europe. And it's probably the foliage of an early seed plant. So there were seed plants, probably more shrubby types of plants growing on floodplains, and that lycopod in the previous, like here, this is sitting adjacent to and above a coal seam. In fact, you can see how coaly even some of these areas are right on the soil horizon. But this is a swamp, just like you find in the upper Carboniferous, all those coal forming forests. So this was happening on a smaller scale in the lower Carbon. There is a coal that was mined in that part of Virginia, near Pulaski, Blacksburg, and all up and down north and south of there, called the Merrimack coal, although it is a high sulfur content coal, so it never was commercially terribly successful. So we think that these lycopods may have formed that coal. But then we have the non-swampy plants adjacent to it, where we have a couple of seeds and 6 different kinds of foliage, and but nothing that looks to us like a fern, sadly. I mean, and it's hard, unless you get the reproductive structures, to know if you have a fern or not. There's also something there that looks like a liverwort. Does anybody know what a liverwort is? I know Jim does.
Yes.
Okay. All right. Now, if you still are with me, I mean, you know, my time seems to be almost up, but I have a few more because I figured, how many of you have collected here in North Carolina? I imagine most of you have collected some plants. Somebody went with me to the Boren Pit years ago. Some of you have gone on your own. You may have gone with Jim or other people. Okay, so I thought I wanted to hit a couple points about this. First of all, I think as a state, we can claim a little bit of fame as having a good array of fossil plants from the upper Triassic period of time here in North Carolina, in the Deep River Basin, and also up in the Dan River Basin. Other places that have decent assemblages of plants in the North America are the United States and Arizona, for example, in the Petrified Forest. And there's a tremendous amount of work that could still be done on fossil plants from there. And the preservation is really quite cool. I saw some things, they've expanded some of the protected land and found new deposits and tons of fossil ferns come out. And these plants collectively provide some ideas about climate. And that generally, this period of time, it was a very warm climate once again. And it appears that at least it was seasonally dry, because a lot of these plants like these kinds of plants have very thick cuticles. Also, some of the conifers that come out have thick cuticles on them. And they're also comparable, for instance, if you have a cycad, to things that can't stand a whole lot of cold. So it's things that are very common and that you find abundantly in, like in what's formerly called the Boren Pit down in the Deep River Basin, would be these Bennettitalian types of leaves, Odoxamites, Xamites, and also this cycad leaf. And you really need cuticles sometimes to be able to tell them apart. So you have to look at the arrangement of the epidermal cells around the stomata. And a stoma is composed of 2 guard cells and an opening, but then there's a group of cells around the outside. and it tells you something about how they developed. And so I put in, you may or may not want to worry about that, but this is one way we tell them. A second is if you have good reproductive structures, you can tell the two apart, but I didn't put those into my slide. Okay, so then there's a number of conifers, and I wanted to show this because I feel this is one of the most exciting finds ever, was finding this big long cone that's several centimeters long. It's called Voltiopsis, and it's probably a precursor to something like pine. But the other thing we have found over the years, and this made my Arizona colleague really happy, although he refused to publish on it. So this is a set of figures of this plant called Dinophyton that occurs in Arizona in the upper Triassic. And he wrote a paper once that said, it only occurs in Arizona, it doesn't occur in Texas and North Carolina. Well, then I did a field trip guide, and somehow the images got to him, and he said, hey, I think you have Dinophyton. And sure enough, when Sid visited, we found out that, yes, I did have Dinophyton. And so this particular weird gymnosperm occurs here in North Carolina as well as in Arizona. Now, the other place you would compare Triassic plants to is Europe, where they have many more exposures of it in Great Britain. And so there's a whole lot that could be said about this, but we don't really have the time. I'm just whetting your appetite here for further investigation. Okay, we also know of a variety of ferns. These are ones from North Carolina. Again, this happened back when they were still doing something in that pit and bulldozing once in a while. I got out some beautiful big chunks of these presumed osmondaceous ferns, like the royal fern or the cinnamon fern today. Here's another one. It's placed in its own family now, Synepterus, which has reticulate venation. This one with these little button-like groups of sporangias called Matonidium. These have been known for quite some time and were described, I think, initially back by Hope and Patterson, and then maybe by, a little bit by some of the other workers. However, I had found scraps of things that I wondered about. And then Brian Axsmith, who has worked on some of this Triassic material through the collections that are now in Kansas. Those are Bob Hope's collections, especially that are at the University of Kansas. And so he's found what's called a filmy fern. So there's a group of ferns, they belong to this family, Hymenophyllaceae, and they have really, really thin leaves. So they're called filmy ferns. And they bear their sporangia in little receptacles at the tips of the leaves, which is exactly what this fossil does. Now, it's pretty amazing to think that one of these very delicate little ferns could be preserved, but it was present, and this material comes from the Boren pit as well. And these are some images from the paper by Brian. A sad note here, we lost Brian to COVID in 2020 early on, which is a real shame because he was doing some very good work. Another paper, though, that he published on, and I think this is interesting, and it's something to keep in mind. It's a lesson to all of us. So most of you are pretty familiar with this fern that occurs in the Boren pit called Pyconopterus, or at least we thought it was a fern, but we didn't know anything about its fertile part, so it was unclear, or just fern question mark. But in going through those collections, Brian found the fertile parts and worked with two other colleagues to publish on this in 2018. So this shows what the fertile portion of a Pyconopterus looks like. If I collected that, I confess I might not have kept it. And you might think it was, well, it was just like, what is this? And just another stem. But if it's well enough preserved, like you see here, and even better here, these little blobs are the clusters of sporangia. So this is actually, if you see the reconstruction, they had, first of all, some foliage of Pyconopterus down at the base of this ugly looking thing right here. But so they think the fertile parts of Pyconopterus were, it's sort of like you see in some of the Osmondaceae today, or Scheisiaceae anemias, for example, that the basal part of you get green photosynthetic leaves, and at the tip end you get smaller leaves loaded with sporangia. So it has a vegetative region and a reproductive region to it. So this is the new view of Pechinopterus, which I think is kind of cool as a new advance on our understanding of Triassic age ferns. So with that, I'm going to end and say, keep hunting. I'm sure I don't have to encourage any of you to do that, but I hope I've also given you a taste of some of the kinds of things that we can do with many of these fossils. I've benefited because some of you have shared information with me about things, and so I'm always happy to come and share what we've learned with you. So thank you very much for having me. I'll take my screen off. Any questions?
Okay, come up to the computer so she can hear. Just stand in front of the screen.
Yes.
Yes.
When did Archaeopteryx become an extinct group? And what were the reasons, possible reasons why it did become extinct?
Okay, so I think the consensus of opinion is that the Archaeopteryx went extinct at the end of the Devonian. And there is evidence for 1 or 2 glacial events happening at the end of the Devonian period of time. There were a couple extinction events that predated the extinction of Archaeopteryx. They were predominantly marine extinction events, the Kelvasser, and I'm drawing a blank on the other one, Hangenberg event. But we see Archaeopteryx right through the top of the Devonian. We do not see it. And there's a place in Pennsylvania where somebody identified something as Archaeopteryx in the lower Carboniferous, but when they did work on the spores, they found that it was latest Devonian. So that's what we draw as a line. And probably climate change was the cause for its extinction.
Thank you.
Other ones?
Any more questions?
Yeah, how do we contact you regarding a fossil piece that was found at the Bohr and Clay mine? It looks like a reproductive organ of a Benedictalian plant.
Ooh, I'd like to see that.
Where I would send photos.
Okay, let me put my email in the chat.
Okay.
So you can send photos to me by email or something like WeTransfer if you need that one, if they're too big to go by email. So just email me, pgensel@bio.unc.edu. And I have one very poor Benedictalian reproductive structure collection, but not too many others. So those are scarce. I don't know how many the museum has, but that'd be interesting to find out.
Well, I'm just an amateur. I'm not positive about the identification, but I'd like you to take a look at it.
I'd be happy to look at it. Other questions?
Victor, this is Jim Nichols. CC me that too. I'd love to look at that too.
Yeah, Jim would be very interested as well.
Sure.
Yeah.
Any others?
All right. How about your opinion about when flowering plants evolved? Was it Triassic, Jurassic, Cretaceous?
Okay, that is a really interesting question, and so what I guess I feel I'm going to be conservative a little bit. So I wouldn't be surprised if at some point in time we found some flowering plants in the late Jurassic. That's as far as I'm willing to go. I don't think things that are occurring in the Triassic are angiospermous. There have been reports of some pollen grains built like angiosperm pollen grains coming from the Triassic, but they lack, when you do ultrastructure on them, they lack a critical layer. And so it looks, so one thing we have a worry about is what we call convergent evolution, where you see a subset of the characters that distinguish a group appearing in another group. So for instance, some of the gymnosperms may look very angiospermous. So for example, these Bennettitalians actually produce their male and female reproductive structures in something that looks like a giant flower. They have bracts, and then they have their microsporangiate structures, and then they have their ovules, which is similar to angiosperms. The trouble is the ovules are not enclosed within a sterile structure like in flowering plants. And then their pollen is very, very different. So, you know, the pollen record has been scrutinized pretty thoroughly. You never know, though, something new can turn up. So I'm willing to say, when we start finding the earliest angiosperms in the basal Cretaceous, that's not the earliest. Those are the ones we're finding. It's just like finding the earliest plants. You keep pushing that back in time, but you've got to be able to demonstrate some of the characters that you think that are conclusive for that group. So that's why somebody else may totally disagree with me, and that's fine, because I think this is what keeps paleobotanists going and paleontologists and so on. That, you know, if you keep pushing, who knows what you're going to find.
All right, thank you. All right, well, thank you.
And thank you.
Any other questions here? No. Well, we want to thank you so much for your wonderful talk today.